So welcome to this webinar of the basic series of EURO-NMD webinars organized with the European Reference Network for Rare Neurological Disorders and the European Academy of Neurology. Today we have with us Stefan Vasilopoulos, one of my teammates. He works at the unit Centre de Recherche in Myologie, Center for Research in Myology of the INSERM at the Pitié-Salpêtrière and in connection with the Sorbonne University. So we are going to have a discussion about morphological and functional issues related to Caveolae and Bin1 from ring-shaped T-tuble initiation platforms. Stefan is a very enthusiastic researcher that takes much pleasure in the work that he does, and he has a very relevant work behind him about triadins and about quaritrin, and leads a group in articulation with other of the PIs in the institute, and I'm sure he will deliver a very interesting and thoughtful presentation. I promise that if no one else puts question at the end, I will put a few clinical questions. So we will need to have always some attempt to translate all those wonderful basic research findings into meaningful ideas for the care of patients. So Stefan, thank you for accepting the invitation, and the floor is yours.
Thank you very much, Antonio. And thank you to the four spectators which are present today. I'm very happy that they were here to be present for this seminar. So it's always really my pleasure, and you're right to say that I'm enthusiastic. I really like this work we're doing in the lab at the moment. And so I'm going to present you this work that has been done for the last couple of years in my group. And it's the main purpose of this project was to understand how caveolae are involved in shaping the muscle cell, and why are caveolae so important that mutations in proteins of the caveolae components cause muscular dystrophies. And so today I'm going to focus on the T-tubules, and this is a very specialized compartment of the muscle cell that I will introduce in a little while. But I just like to first say that we are in the lab. I have been interested for several years in all the different aspects of the membrane trafficking, and how membrane trafficking machinery is involved in not only the physiology of the muscle, so how the muscle membrane trafficking machinery allows the compartmentalization of specific proteins, and how they are involved in the basic formation of the muscle, but also to understand why so many proteins involved in membrane trafficking are mutated in muscular dystrophies and in congenital myopathies. And there are several compartments which are of interest in the muscle cell. Today, I'm going to focus on this specific compartment, which is called the T-tubule. And these T-tubules are invaginations of the plasma membrane. But what is more important is that mutations in proteins which are involved in the formation of these T-tubules or in their function have been associated with a number of congenital myopathies and muscular dystrophies, and these are just the beginning of the list, because T-tubule malfunction and T-tubule defects are involved in other muscular dystrophies which are not directly related to proteins that are components of these T-tubules. So, for instance, there are subsets of muscular dystrophies which have been involved in or related to defects in the sarcolemma, but which also have a component of the T-tubule system. And so, very briefly, this is just a kind of scheme you will find in the internet. What is a T-tubule? Well, usually the T-tubules don't come alone. They come with another component called the sarcoplasmic reticulum, and in fact, this part of the sarcoplasmic reticulum called terminal cisternae. And the T-tubules are actual invaginations of the sarcolemma which penetrate in depth in the muscle fiber and which allow a mechanism called excitation contraction coupling. So very briefly, when an action potential is transmitted from the motor neuron to the muscle fiber, this action potential is going to propagate along the sarcolemma, but will then penetrate inside invaginations of the sarcolemma called these T-tubules. And there, in these T-tubules, you have the localization of a calcium channel called the DHPR. This is a voltage-sensitive calcium channel. And the particularity of these structures is that they are directly opposed by these sarcoplasmic reticulum calcium stores, which are in fact covered by these ryanodine receptor proteins, which directly interact with the dehydropyridine receptors. So when this action potential comes, it will change the conformation, produce a change in the conformation of the DHPR, which will in turn activate the ryanodine receptor and cause the release of calcium into the cytoplasm. So this is a very fundamental mechanism, and it has taken researchers quite a while to figure out exactly how this works. So it turns out that you have a triad, which is a structure composed of one T-tubule, and it's opposed by two terminal cisternae of the sarcoplasmic reticulum. And each T-tubule contains the DHPR, and each sarcoplasmic reticulum cisternae on the sides contain the ryanodine receptor. And this structure forms a triad. So if you look to a triad by confocal microscopy, which is not as resolved as electron microscopy, one triad will correspond to one dot in the picture that you see here, where a longitudinal muscle section was labeled with antibodies against the ryanodine receptor, so the calcium channel that is localized in the sarcoplasmic reticulum. In these pictures, you wouldn't be able to distinguish a ryanodine receptor for a DHPR because one triad corresponds to one dot, and there are two dots here because there are two triads per sarcomere in the mammalian muscle. So what is interesting is that the initial experiments aiming to understand in cultured cells how are these T-tubules formed initially hypothesized that these T-tubules were resembling the ultrastructure of caveolae, and they seem to emanate from the surface of the cell, from the sarcolemma, in regions that were enriched in caveolae and which were forming these honeycomb-like structures, like the one you see here. And this is something that was confirmed by Rob Parton's group that showed that in muscle cells in culture, you have an association of caveolae regions with developing T-tubules in structures which look like this. They were regions where you have a small accumulations of caveolae, which seem to be the regions from where T-tubules would emanate. And in fact, this was substantiated by work performed by Clara Franzini Armstrong, which is a real expert, world expert into the T-tubule and the triad field, who also showed, while she was in sabbatical in our lab in Paris, that caveolae can accumulate in mammalian muscle. This is mouse longitudinal sections observed by electron microscopy. And what you see here is that the caveolae accumulate at the neck of these T-tubules, and you have these large accumulations of caveolae. So it seems as if there is a link between accumulation of caveolae on the surface and formation of T-tubules during the development. So what are these caveolae structures? Well, caveolae are invaginations, small invaginations, like small caves, cave-like invaginations of the sarcolemma. They are almost 60 to 70 nanometer in diameter, and they're initially characterized by George Beladi in the 50s, who was the first one to describe these structures and who was the first one to describe this striped structure of these caveolae coat. So if you look at them very carefully, you will see that caveolae present these small striations at their surface. And they're always the same. If you make longitudinal cell sections, you will see these accumulations of caveolae at the surface. They are what you call omega-shaped structures. Now, if you look at them by another electron microscopy technique, which is platinum replica electron microscopy, you see that these caveolae structures are composed of a coat. That means that it's not just membrane, there is a protein there. This protein either forms these spirals. In fact, it was initially thought that caveolins should be called spiralins. But it turns out that most of the time, these caveolae proteins are localized in stripes at the surface of the caveolae. So this is an important feature because always every single cell has caveolae, and every single caveolae is composed of these little stripes. And the second thing which is important is that these caveolae are very sensitive to cholesterol levels. So this was shown by John Heuser himself. He was doing these experiments by treating the cells with methyl beta-cyclodextrins or statins, and he would see them completely disassemble or completely flatten down on the membrane. So the caveolins are integral membrane components of these caveolae. And there is a specific isoform of caveolins in muscle, which is a caveolin 3 isoform, and it's expressed in skeletal muscle and heart muscle, and it has been shown that caveolin 3 knockout mice present defects in the formation of the T-tubules, and then if you perform electron microscopy sections on muscle from caveolin 3 knockouts, where the T-tubules have been labeled with a dye, like that you see here, that you would see that they no longer have these almost crystalline organization, but they start having a different orientation. So they are turning longitudinal when normally all the T-tubules should be localized transversely in a longitudinal muscle section. So this kind of put a hint that these caveolins were important for the formation of the T-tubules during developmental steps. And in fact, they're not only involved in the T-tubule formation, they seem to be involved in maintaining structural integrity of muscle. This was something that was proposed quite a while in the beginning of our understanding of caveolins, that more recently have been they have been connected with mechanoprotection. So cells that are no longer capable of repairing their membrane they need caveolins to actually repair the membranes. They're involved in the calcium homeostasis. And last but not least, they seem to have some role in the formation of T-tubules, although this is unknown. However, why this is important is because there's a whole family of protein of myopathies, limb-girdle muscular dystrophy 1C, which are called caveolinopathies and are caused by mutations in either caveolin 3 or another component of caveolae, which is also a muscle-specific isoform. This is the cavin 4, also known as Merck protein. And so both of these proteins, which are muscle-specific, can be mutated in caveolinopathies, which are characterized by, so the severe cases present with a limb-girdle muscular dystrophy. It's no longer classified as limb-girdle muscular dystrophy 1C, but it is some cases of caveolin mutations can cause quite a severe muscular dystrophy. There is another aspect of these caveolinopathies, which involves this rippling muscle disease, and this rippling effect is caused by the hyperexcitability of the muscle, and it's involving contractions without having action potentials measured. So it's these silent action potentials. One really strong characteristic of these caveolinopathies is that they are mild, but that they can be detected by the high CK levels in the blood of patients with mutations in caveolin 3 or cavin 4, and some different forms of caveolinopathies involve distal myopathies. What is important also to know from a genotype-phenotype correlation is that there is no direct connection between the mutations in the caveolin 3 and the phenotype of the patient. So it hasn't been able to be shown that some mutations are responsible for the rippling effect, or some mutations are responsible for strong, severe limb-girdle muscular dystrophy. And so there are several phenotypes which are associated with mutations. The first one is that caveolin 3 is no longer targeted to the sarcolemma. Well, in fact, I will show you later on this presentation that it still is, but a large proportion of the caveolin 3 is retained in the Golgi. It has been proposed that these myotubes or muscles from patients with caveolinopathies have a membrane fragility. The T-tubule defects have been quite well shown before, and also that there is a calcium defect with the DHPR. So again, a defect in the T-tubule function, not the formation, but function this time. And before I start showing you the results, I want to show you a last introduction on a protein that is also really important for this project. It's a protein called amphiphysin 2 or Bin1. This protein is expressed throughout the body, but in skeletal muscle, there is a specific splicing of this Bin1 protein, which leads to the incorporation of an exon 11, which is involved in for tubulating the membranes. But more importantly, Bin1 mutations have been shown to cause centronuclear myopathy, and also that they cause T-tubule defects in centronuclear myopathy patients. This was shown elegantly by the group of Jocelyn Laporte, but it was also shown that Bin1 splicing of the specific exon 11, which is involved in forming T-tubules, is also abnormally spliced in the myotonic dystrophy, which is a spliceopathy characterized by alternative splicing defects, and especially involving the ones that occur during the maturation of the muscle from the embryonic to mature to an adult state. So again, pointing to a really important role for this Bin1 in forming these T-tubules. So the question that we wanted to ask, and I say we because this work was done with a PhD student, Eline Lemerle, at the time in the lab. And the question we wanted to ask was, how are these T-tubules formed? Because we know who contributes to this process, Bin1, the calcium channels, and other proteins, but we really don't know how and when during development do these T-tubules form, and what is the mechanism. The only thing we know is that it seems that caveolae, either single caveolae or accumulations caveolae to plasma membrane, are initial steps for the formation of the T-tubules, and that at some point Bin1 comes in and tubulates the membrane like crazy, because this protein is capable of producing really long tubules, but we still don't know at what point does the sarcoplasmic reticulum connect with the tubule, and what does the cytoskeleton do during this process. So what we really wanted to revisit was the mechanistic aspect of how our T-tubules form in muscle. But to do this, we needed to change some things. So if you keep on doing the same experiments, but it will not produce any new knowledge. In this case, what we wanted to do was to combine electron microscopy and, in fact, correlative electron microscopy to the analysis of the T-tubule in muscle cells, which have been extensively differentiated. So the first set of experiments that we do is we perform this platinum replica electron microscopy, where we unroof cells using the sonicator, and we can then produce a platinum replica of their surface, which is capable of going under the electron beam in an electron microscope. So this allows us to have the highest resolution. We can observe the objects in 3D, and I will go in more details along the talk. What if you do this, if you unroof cells with sonicator and you label caveolin 3, you only have the ventral membranes left. So this is where caveolae are localized. So the labeling is very good and the accessibility is very good. And if you then look at them at the electron microscopy level, you see that you can clearly observe the beautiful coat, the striped coat that caveolae present at the surface, but you can also have a very high resolution, as you can distinguishing between either flat caveolae, such as this one, or fully budded caveolae, such as the ones you see in the picture. And when we did this, and we did this in our muscle cells, we saw these rosettes that I mentioned before, these large accumulations of caveolae together, and we also saw some quite interesting structures, such as this one here, in regions where the plasma membrane is completely occupied by caveolin 3 caveolae. They start shaping the membrane and are capable of deforming it in ways that we were not used to seeing in other cell types. But the cells we were using were not differentiated enough. So the second point that I think is crucial in this experiment is to actually differentiate the cells far enough so that they can start forming T-tubules, because this is really a limiting step in the formation of T-tubules. Is like the neuromuscular junction. If you're not well mature enough, then these structures don't form. So in this case, what we did is that we influenced our protocols from the one that Edgar Gomez was doing while he was in the institute a few years ago, and we differentiate cells. These are primary cells, primary myotubes from mouse. We differentiate them. And then once they form these myotubes, we cover them with matrigel. So it's almost like an organoid-like culture, so that these muscles don't detach from contractions. And we also supplement with a differentiation cocktail, including agrin, which is also pushing towards a higher differentiation of these myotubes. And what is interesting, so if you look at these two proteins that I mentioned before, the DHPR and the ryanodine receptor calcium channels, when you do these experiments for 5 days of culture, then both DHPR and ryanodine receptor are overlapping in these sort of cluster-like structures. You see here, they are not particularly organized except for maybe some longitudinal organization. If you do the same experiment, but now you let this myotubes differentiate for 9 or 10 days with this matrigel and with this supplementation with agrin, now these triads are starting to organize themselves, and the DHPR and the ryanodine receptor are both forming these double rows of dots. So this is like almost like a proxy for high differentiation of these compartments. So we did these experiments. We unroofed cells that have been extensively differentiated, and the first thing, and labeled them with caveolin-3 antibodies, or expressing caveolin-3 GFP to have just a omit the necessity to label the caveolin-3. And what we noticed is that caveolin-3 was always localized in these forming these circles, these rings on the ventral side of the membrane of cultured myotubes. So to prove it, we decided we were going to use correlative EM, and we would try to overlay the fluorescence with the electron microscopy signal. So in this case, we cultured cells, we unroofed them with a sonicator, but before producing the replicas, we labeled them with antibodies, or we look at them by high super resolution light microscopy. And when we do this, we use these glass cover slips, which have these all these alphanumerical graduations on it. And that allows us to trace the same cell that we have just imaged with super resolution, but then we can trace it all the way on the replica, and we can overlay the labeling for the caveolin-3, and we can see what are the structures that are labeled by these antibodies. And so, of course, the question was, what were these rings caused by? And to no surprise, we found that these rings were formed by a circular assembly of caveolae. In this case, you have a circular assembly, but the labeling for the light microscopy is patchy, and you can see it. But sometimes, if the resolution is good, then you can see a circle, and then that same circle can be overlaid on top of a ring of caveolae. So it seems that the muscle cells are pushing the caveolae towards forming these circles at their surface. And that was very interesting, because the more we did, the more we saw them. And suddenly we just had all this time, every time we could look at the electron microscope, we saw that caveolae were forming these circular structures. You can see that the striped surface of the caveolae are still visible in these rings. And then we have a plethora of different examples. We have naked rings, and then we have these rings with the caveolae, still the stripes are visible on the periphery, but then you have all these caveolae accumulating on one of the poles of the ring. And even more surprisingly, we had these rings, and then we had these membrane tubes that were reaching out at the circle at the center of the ring. So this was very interesting. What are these? And so, to make the long story short, these structures, tubular structures that are reaching out and forming almost like a synapse with the center of these caveolin rings, were nothing less than cortical endoplasmic reticulum, which is like sarcoplasmic reticulum is always there in these preparations. We always see it. We weren't expecting to have such an intimate interaction with these caveolin rings. You have one here with these, every time the SR is colored in purple here, you have another one here and another one here. In this example, you have a double ring, which is all surrounded by these ER tubes, sorry. And another long here, you have a long caveolae ring with the ER surrounding it. So although this is not a perfect association between the two, we see that whenever we have these caveolae rings, they are surrounded by these ER-SR membrane compartments. And so what you see here is one of the initial attempts to do the correlative visualization of the rings. in regard to the ryanodine receptor labeling, which is a marker for the sarcoplasmic reticulum membranes. So you have initially, in the first pictures, you will see the light microscopy, and then we'll switch to the EM, and then you have higher magnification of this electron microscopy, and again, the correlative, where the overlay between the two channels. So here is one ring, and then if you look on the electron microscopy, you can overlay the signal for the light microscopy. Here is that same ring that I showed you before, and you can zoom into it, and then you can see the actual ring formed by these caveolae. This is just a tomogram showing the 3D, and then this is the ryanodine receptor, which is labeling the ER tubes which are surrounding the ring. I can show it again. I love this movie. Since we're not too many, I can have fun. And oh, by the way, please interrupt me. I would be happy to take any questions in the meantime while I'm talking. Okay, so this is interesting because it shows that these rings are physiological. We can localize them. We know that they are there. Now the question is, what role do they have in this T-tubule formation? So the first question we wanted to know was, okay, the ryanodine receptor sarcoplasmic reticulum triads are not too far away, but where is the DHPR, the calcium channel that goes into the T-tubule? And so we did this experiment. We used the lentivirus that expresses the alpha-1s subunit of the DHPR calcium channel. It's tagged with a GFP tag on it. And then we culture these cells, we unroof them, and then we label the caveolin 3. And what was fascinating is that, but that was something that had already been shown by the groups in Lyon, which are the electrophysiologists, which had already shown that there was a link between caveolin 3 and the DHPR calcium channel. But it was very satisfying to see such a nice colocalization on the light microscopy. And so then when we went on the electron microscopy and we did these CLAM experiments, it was beautiful to see that the DHPR calcium channel very early on during the formation of these T-tubules is already accumulating in these caveolin-rich regions on the sarcolemma. And you see there is a quite striking colocalization between alpha-1s and caveolin 3. So this was a good point, was when are these T-tubules formed? Can we see the T-tubule? Because I have talked a lot about caveolae. I showed you the ER tubes, but I haven't shown you the T-tubules yet. So this was the next question we wanted to see. Can we trace the T-tubules? Do we need to always use a protein marker? Can we use a lipid marker to look at these T-tubules? And do we know when do they start and when do they end? So to do this, we use two tools. The first one, and still every experiment I'm talking about now is on these extremely differentiated myotubes that we are letting go for 10 days in culture with the matrigel and then the agarine. Now, we found this dye called Membrite, which is a dye for live cell imaging, and which binds to lipids on the outer bilayer. So if you put this membrane dye in live cells, it's in the first few seconds, it's only going to label the plasma membrane. But if you leave it for a few minutes, then because it's not going to be endocytosed, very quickly it only can diffuse into the T-tubules, which are connected to the plasma membrane, and they are open to the plasma membrane. And if you leave these membrane dye with the myotube for a few minutes, then before it has time to endocytose, it's going to diffuse in these tubes, and you actually see these tubes lighting up as you put the membrane dye. And after a while, these cells are going to take it up by endocytosis, and they are going to have their lysosomes, their endosomes, everything's going to be full in membrane, and they're going to die. And because it has some phototoxicity. But this assay was great because in a few minutes, you could label on live cells the whole T-tubule system and know exactly where the T-tubules start, where do they go, and what you see is that they are always starting from the plasma membrane and they're bifurcating like this and become longitudinal. And as you see here, the majority of the T-tubules in our differentiated muscle cells are not transversal yet, they are still longitudinal. So we did these experiments, and then we can we can image them and make movies. And we had two tools to look at them. So Membrite is a tool to look at the lipids that are composing the, or because it binds the lipids of the T-tubule. We also wanted a protein dye. And in this case, we took the obvious candidate, which is Bin1. So if you label Bin1, it labels exactly the same tubules that you label for Membrite. And what was interesting is that both Bin1, you see here, and both Membrite were showing up rings at the surface of the muscle cell. So these are images that are taken with a confocal microscope on the bottom of the dish, and you see that the Membrite is labeling these tubules, which are always forming these rings. There are several of them in this example. Each one, there's always a ring whenever there's a tubule. And the same thing with Bin1. Bin1 is doing exactly the same thing, is labeling the same network. So what is going on here? We kept on doing these experiments. We thought we're going to find a solution because we have all the right tools here to understand what's going on. So we repeated and again, and we could always see the rings made of the caveolae. And by doing it a lot, then we started seeing tubes that were coming out of these caveolae rings. So here is one that I like a lot because the tube is just starting to form. And here is another one when you can even see the caveolae material leaving from the ring. And this one, which again shows this almost spherical structure and this longer tubule sticking out of it with these beaded structures. So the caveolae are localized all along these tubules, and they're forming like a beaded necklace. These pictures were the first ones that gave us the idea that this could be a potential mechanism, that you didn't just make a tube leaving from the sarcolemma out of one caveolae going drrrt, but you need to form a platform of caveolae assembling in a ring, and that only then could you start forming tubules from these cells. And these examples were confirmed really elegantly by my colleague Jeanne Lenné, who is an expert in thin section electron microscopy. So this is just another way of doing this. Is more of a conventional electron microscopy technique. And what she showed by doing very thin sections right on the part where the cell was adhering with the substrate, so this is called the ventral part of the membrane, she was able to show that you have these circular structures with full of caveolae around them and these long tubules which should emanate from them. And here, why I like this picture is because you see it connecting with an endosome or endolysosomal compartment here. This is late endosome, which is connecting with this T-tubule. And in fact, you might even think that there could be some lipid exchange between the endosomes and the T-tubules, because you have to imagine that to elongate these T-tubules, you need a lot of lipids. So maybe these lipids are transferring from the endosome to the T-tubule, and this is helping the T-tubule grow. But anyway, to see this was a very good confirmation that it was just not just an artifact of the replica technique. So not only is not an artifact, but we were able to do it by correlative EM. So we labeled caveolin 3 and Bin1, and you see that if you unroof these really differentiated cells, you start seeing these rings, and from which these tubes made of pearled tubes made out of caveolae are emanating from them. This is one example, another one, and another one here, where you have one spherical structure here, another spherical structure, and it's all connected by the tubes, which are positive for Bin1. So this is a lot of cool electron microscopy. It's always what help us figure out what's going on, but we now wanted to go to another step, which is to test how functional we could analyze this experiment, this situation, this mechanism. So we did the experiment where we overexpress Bin1 in cells, but we express the isoform which lacks the capacity to tubulate. It's lacking this exon 11, or we overexpress the exon 11 expressing Bin1. And what you see here, and what we quantified later, is that the overexpression of amphiphysin 2 Bin1 without exon 11 is not forming any rings, while the one with exon 11 is forming these rings. What was really interesting was that both constructs were actually capable of forming rings. but this one is not capable of forming tubules, and this one is. So both amphiphyzins, both bin1 isoforms are capable of forming these rings, but only the one that is the muscle-specific isoform is, because has this extra exon 11, is capable of elongating these tubules. And so to go back to the caveolin 3 business, so if you take your cells, you transduce them with this bin1. Now we're using this as a tubulation assay. We're overexpressing bin1, and we're seeing, is the cell capable of forming tubules or not? Now, if you take control cells and you express the bin1 isoform, which has exon 11, it's going to make tubules everywhere. It's going to tubulate and form rings and tubules everywhere. But if you knock down caveolin 3, now the T-tubules are very strongly reduced. So in this case, you see there are still some tubes formed, but they are much less than in the control situation. And see here, you see the efficiency of the caveolin 3 depletion, which is quite good, which suggests that caveolin 3 is important for this tubulation process. So we turned out to bin1 to understand exactly why was this happening. And so here you see I have an example of bin1 overexpression through our adenovirus system in cells in unroofed and myotubes, and then the myotubes are unroofed. And the first thing you notice is not very different from the caveolin thing, is that you see rings. The bin1 is forming tubules, okay, a lot, but it's also forming rings. So here is an example that I like a lot, because you have this central ring, and then you have all these tubules sticking out of it, just like what we were seeing for caveolin. And then once you see one, then you see them all. There's always, bin1 is always forming rings from which tubules are seem to emanate. So we really needed to confirm these data by another technique than electron microscopy. And the best thing we could do, and for this we collaborated with Bruno Cadeau in the lab, we did live imaging using our super resolution system by either tagging caveolin 3 and following caveolin 3 in differentiated myotubes, or labeling bin1 and following bin1. Now in the case of caveolin 3, you see a lot of rings, but not a lot of tubes. And then suddenly you can see a tube just there is going to come out of this. You see it here. This is always the same case. You see the ring, and then at some point you see the tubes. With bin1, when you overexpress bin1, it has such a strong tubulating capacity that the whole cell is full of tubes and rings. And so here it was much easier to find it. You have this ring, it forms, and then the tube leaves. And the same example here, and this one, I like it a lot as well, because you have one ring, but then you have two tubes leaving, or even three rings leaving from the same tube. So to us, this was a real confirmation that this wasn't really not just in our heads, but this was a physiological mechanism that was relevant. We went on to do exactly the same experiment by overexpressing bin1, but this time we looked at it by electron microscopy. We either unroofed the cells and we see the structures formed by bin1, which is actually tubulating caveolae, because this is full of caveolae-like structures. But we also saw it from the outside without unroofing the cell. So here you do nothing, and you just metal coat the cells and look at them by electron microscopy, and you see that all these holes correspond to caveolae, because caveolae are invaginations which are connected to the outside. So you see all these holes, which are the caveolae, which are assembling into a ring-like structure, and from which some tubules seem to elongate. So to prove it, and to understand who was coming first, is it caveolin that form a ring and then bin1 tubulates, or is it bin1 forming a ring and then caveolins are attracted to this ring and then tubulates? And in fact, the answer is the second possibility. And the reason we think it's the case, it's because in very in situations where we overexpress bin1 in cells, we have cases where bin1 forms these ring-like structures, but there are no caveolae on them. So bin1, and so you have this one example here. Sorry, you have one example. This is the case here where you have this ring. It's a characteristic, but the ring in the electron microscopy doesn't show any caveolae or any stripes on it. So this is suggesting that you can have a ring without the caveolins. And the same thing here, we have this double ring, which has absolutely no caveolae around it, but it's just composed exclusively of bin1 overexpressed in cells. And if you do this in primary cells, which are the most elegant way to differentiate them, they are the system which for us drives the most, the further in the differentiation, but this time we transduce them with the bin1 GFP, you see that bin1, there's a green here, there's a mistake here. This is caveolin 3 and this is bin1. Bin1 is capable of forming these rings with a very good resolution. You see that bin1 is localized everywhere on the ring, but the caveolin 3, which is also enriched on the ring, is a little more diffuse, suggesting that first comes bin1, which forms the structure, and then it accumulates caveolae on the structure. And here, this is a tomogram that's corresponding to this correlative EM here. You see the tomogram of the ring and the tube emanating, which is positive for bin1 in green and caveolin in red. Okay, and the last piece of data for this purely basic part of the presentation, basic science part of the presentation, is we collaborated with Laura Picas and Montpellier to prove that this was something that was capable of happening in vitro, that bin1 is capable on itself to form a ring. And so what Laura did is that she either incubated bin1 with phosphoinositides on a supported lipid bilayer, which was either phosphoinositide PIP2 or PIP4. And she saw that with both phosphoinositides, but especially with this PIP2, bin1 was capable of assembling completely in vitro. There is no caveolin in this case, assembling into these circular skeleton-like protein structures, assemblies, from which these tubes emanate and scaffolds emanate. And there's very characteristic that there is always one central tube, and there are all these side tubes, scaffolds, which are shorter than the longer one in the middle. And next to it, I have shown you the pictures from before, from the in vivo experiments or in cellular experiments, where you overexpress bin1 and unroof the cells. And you see that there is a very striking resemblance between the type of structures that bin1 is able to form in vitro and the types of structures that bin1 is capable of forming with caveolins in cellulo. Okay, so just to wrap up this part of the talk, and then I'm almost done here. What we think we discovered is that bin1 is, by its bar domain, this protein is like a banana-shaped bar domain, which is able to pinch the membrane. We think it's capable of pinching the membrane in a circular fashion, and that these circular domains attract caveolae, that the caveolae accumulate along the ring, and then as further caveolae or more caveolae come along, they polarized at one of the poles of these rings, and that at some point, recruitment of bin1 on these structures, or additional recruitment of bin1 on the structure, leads to the tubulation and to the elongation of a tube. And these tubes ultimately connect with the sarcoplasmic reticulum terminal cisternae containing the ryanodine receptor and start forming the early couplings that are important for the maturation of the muscle cell to reach a degree where the cell will be able to form maturity tubules. And that last step we think is important because if you want to make a connection between the DHPR and the ryanodine receptor, then you probably need to remove the caveolins from the tube, and then these caveolins need to recycle or to diffuse along the tube for the contacts between the DHPR and the ryanodine receptor to start forming. And the last part of the talk that I will present is concerning the caveolinopathy patient myotubes. So we decided to do this exact same analysis, but this time, instead of using control cells, we used immortalized myotubes from patients with different caveolin mutations. The experiments I'm going to show here are both two patients with the same R26Q mutation. The mutation is here. It's in the oligomerization domain of caveolin 3, and the phenotype of these patients is at the cellular level, a decreased caveolin 3 level at the plasma membrane, less caveolae at the sarcolemma, and an accumulation of caveolin 3 at the Golgi of these patients. And this mutation is associated with the most severe cases of limb-girdle muscular dystrophy 1C form, with rippling disease and with high CK levels. So the first thing we did is we tried this tubulation assay. We cultured control cells or these caveolin 3 mutant cells, and we transduced them with the AAV expressing GFP bin1. And what you see here, as expected in the control situation, caveolin 3 positive, healthy myotubes overexpress bin1, start forming these tubes everywhere, like small little hair everywhere and rings. But the myotubes which are expressing the patient myotubes were not capable of forming these tubules. So what you see here is the quantification of the T-tubules. If you have no exon 11 in bin1, you form no tubules. If you have exon 11, you start forming tubules, but in a mutant patient myotubes, you have a strong decrease in the amount of tubules formed. And if you measure the rings formed in these myotubes, where they're much less, or at least almost half of the rings that you see in a controlled situation. So, and we further characterize the ultrastructure of these myotubes to see, can we detect rings? Can we see how the caveolae are? So the first thing is that the caveolae have an altered morphology. The rings are very rare. These are the few instances we found was this example here, where there is no tubule emanating, and it doesn't seem to be a normal ring. It's smaller. They have these small accumulations of caveolae here, or this huge one, where you see we pseudo-colored the caveolae, which are forming surrounding the tube. But to make this long story short, in majority of the cases, what we saw in the caveolinopathy patients were these small tubules, which seem to be made of the concatenation of several caveolae put one next to the other. And this is something, in this example, there are several, but we have many, many examples of small little tubes like this, instead of forming rings and tubes emanating from the rings. And so this is also something that we quantified. If you overexpress, if you have the control situation, you have these rings forming at the surface of the myotube. If you are in the mutant cells, then you have a decrease. You still have a few rings, but you have less rings than what you would form in the healthy myotubes. And so we decided to show this by rescuing this phenotype, by expressing or overexpressing caveolin 3 in the myotubes from the patients. And so when we do this, then we start forming these rings again at the surface of the muscle cell, and we can rescue even not only the number of rings, but also the size of these rings. So as I mentioned in the previous example, you have these large structures. If you quantify the rings in the patient myotubes, you have an increased diameter of the rings in the caveolinopathy patient myotubes. But if you overexpress caveolin 3 and you bring back the full length protein, then you can rescue the formation of these rings at the surface. And to finish, we also analyzed the muscle from the patients in muscle biopsies. This is an example from a caveolinopathy patient with a specific caveolin 3 mutation. He has not a very severe phenotype. What I can say is that he has some centrally nucleated, centrally positioned nuclei. He has also a few invaginated nuclei, and some cases we see instances of very light core-like lesions on the center of the fiber that you can see with oxidative coloring. But at the electron microscopy level, again, the phenotype is not so severe. What we've noticed is that with the caveolinopathy patients, we often have dilated longitudinally organized tubules and dilated tubules. They are a little bit larger than what they should be in the control situation. But what we noticed, which was more interesting, is that, but there are rare instances, that in some rare instances, you see these T-tubules which are bifurcating. So instead of forming a line, they are forming these Y-shaped structures. And these are two of the examples you see here. They are bona fide T-tubules because you can see the contacts they make with the sarcoplasmic reticulum. Here you have the ryanodine receptor feet, which are very characteristic. but the fact that he's forming a Y is something that you do not see in a controlled situation. And this is a really beautiful tomogram made by Clémence Labasse at the Risler Pavilion, where she shows, actually, she demonstrates that it's an actual bifurcation of the same tubule, and that these are not just two T-tubules going along each other. And I think I'm finished with this. I'm maybe 5 minutes late, but I just take another minute to thank all the team, especially Eline Lemerle, who did all her PhD on this project, and Jeanne Lenné, who did all the thin section electron microscopy on this project, Bruno Cadeau, who helped me for the light microscopy, the neuromuscular morphology unit, and especially Clémence, who did a lot of the EM. Our collaborators who are also the lab where I did my PhD under Isabelle Marty's supervision in 2003 at the Grenoble Institute of Neuroscience, who are experts in the EC coupling field, the MyoLine platform at their institute because of all the cell lines that they generated, and all our collaborators. And thank you for your attention.
Thank you very much, Stefan. Pretty arrogant presentation as usual, and I was amazed by your movies, because you start blending the lines between morphology and physiology. We see things moving, and it gives us an idea that these processes are not so static or so solid and rigid as they may seem when we look at photos. So I adored it. So I need to be careful with what I'm going to ask you, because there are no questions at the moment, because we have very distinguished presences in our audience. But as I told you before, one of the things I would like you to speculate on is a bit of how your work and these amazing discoveries fit in some aspects of the diseases. I understand that probably we cannot give, or you cannot give a definite answer, but I would like to challenge you to talk a bit about the rippling and the relation to the phenomenon that you've just described. And also another thing from the clinics is that frequently patients with Caveolae disease complain much more frequently about pain than others with other muscular dystrophy conditions. So I would like to know your thoughts about it, and I'm going to mute all the audience so that people may participate. For instance, Norma is on the line, Norma Romero, and I would like to know her opinion on the same talk, on the same subject as well, if she doesn't mind.
Hello, Norma. Okay, so I like this question because it touches on something that unfortunately there is no explanation yet, or at least no clear explanation mechanistically speaking in my head. It's very interesting that it turns out that the rippling disease, the rippling of muscle disease, or the rippling effect concerns some sort of hyperexcitability of the muscle. So and you noticed I mentioned that these DHPR calcium channels are accumulating in the caveolae rich regions in the controls, and I'm almost sure, because what characterizes this effect is that you have these silent action potentials that lead to contractions, but you cannot measure an action potential. So it smells as if there was something involving the DHPR in there, not a caveolin, a channelopathy per se, but that the fact that maybe part of the DHPR is recruited, but not all of it, because of the caveolin mutation, could lead to maybe some very subtle action potentials which are hard to measure, but which can lead to contractions and which can produce this phenotype.
And am I wrong to presume that this is just the interface between the two sides of the excitation contraction coupling, because we have a kind of sodium-potassium slope from the nerve to a more calcium-dependent activity from the muscle, yeah. And it's something that blends a bit in there as well, doesn't it?
Yeah. Absolutely. It's what I just cannot understand is maybe it would be nice to have this conversation with electrophysiologists to understand how or what do you need in terms of the electrical function to have these rippling effects, because it's very specific. It's almost as if there was this shivering, and then sometimes you can have these. It's called the percussion caused effect, and when you just have a very strong mechanical hit on the muscle, and that completely causes this rippling effect. So it's very interesting because it mixes the mechanical aspect and the electrophysiology. The caveolae are really important for these two things, because on one hand, they have been shown to be able to flatten and mechanoprotect the sarcolemma by their flattening. And on the other hand, they have been important in calcium homeostasis and this AC coupling. And I'll just say one last thing because I think it's relevant, but I have no proof. These bifurcations are very bad. If you have a bifurcation in your T-tubule, the action potential is going to separate in two and it's going to fade away instead of reaching the core of the fiber. And so if you are always dividing the potential, then it's losing its strength. So maybe this is also related to it.
What about the pain?
The pain, well, the pain is something that is characteristic for, I think, all the patients that with caveolinopathies, they have these cramps. So I've heard of cases from neurologists where the only reason why they did these genetic analysis on a patient was because he was complaining of myalgias and cramps. And then they saw that he had a high CK, so they thought, maybe there is some mutation in there, and they found the caveolinopathy. So this is not anecdotic. I've heard several instances where it was the pain and the cramps that conducted for the genetic screening. That's also very, very relevant for these calcium defects, the cramps, because it means this, for me, this hyperexcitability of the muscle. Either you have it for the rippling or you have it for the cramps, is a little bit the same, is that these muscles have the tendency to spontaneously contract, or contract with very little action potential behind it.
Excellent. I was wondering if Norma would want to unmute herself and give us some ideas of her own from her experience, and Bigot is also on the list, so anyone can do the same if they want.
I think this is a topic that is difficult to extrapolate too much on, because even I, when you mentioned this rippling muscle disease, I have not gotten myself around exactly where is this coming from. It's difficult, and in fact, because it's atypical, that even the literature is very poor around this topic.
Yeah, so that's good news, because you have lots of work to do.
Exactly.
So thank you,
Right. Thank you, Antonio, for the invitation. It was
very interesting, and you will sure have many hits and reviews during the phase where we make available the recording on the website.
Sure. And I would be happy to do again, for what Teresina asked, to make a presentation of more generally everything EM.
Yes. Thank you very much.
Thank you, everybody, for being here. It was a pleasure. Bye-bye.
Bye-bye.